This is an app that I developed for a company called Canurta Therapeutics in order to help them bring their medicines to market as fast as possible. This is the DEG pipeline assistant, and what it does is it takes normalized RNA sequencing data and outputs a differentially expressed genes and pathways across conditions so that you can see the biological effects of a drug being tested. RNA sequencing is a common method used in preclinical trials, so testing on model organisms before moving on to humans. Companies who outsource the sequencing job to a third-party lab run into a bottleneck when they reach the downstream bioinformatics analysis after receiving the initial data from the lab. And so I developed this app with Canurta's bioinformatics team in order to better automate this process. So now let me show you how someone would actually use this pipeline. So the first parameter here is the normalized data file, and I'm just going to navigate to that file and upload it here. And then the second parameter are the pairwise comparison files, so I'll upload all of those now as well. And while that's uploading, I'll tell you that these two sets of files are typically prepared by the lab doing the actual sequencing, so these files will be on hand for whoever's using this tool. So now once those upload, the next parameter you can see are the conditions. So these are the conditions of the actual experiment, and I'll put in the ones that Canurta used for an experiment they did with zebrafish induced with ALS-like symptoms. So I'll add those in there right now. And there they are. And one thing that's really cool about this app is that the first layer where all these parameters get sent integrates ChatGPT through an API call. So you don't have to use exact syntax with quotes and commas. I can write something like "here they are" ahead of all these conditions, and I'll show you that that'll be transformed properly to create the command. So the number of replicates, they use 6. And with the natural language, that same thing also applies here. So with the model organism, Dririo is the actual code for zebrafish that would be required for ortholog mapping with gprofiler. But what we can do is just write zebrafish, the common organism name, and that'll be understood just fine. Next are the pairwise comparisons of interest. So a lot of times you're not going to be interested in all the different comparisons generated. Here are the ones that Canurta was interested in for their study. Now, these next 3 boxes here are technical parameters for the analysis, and so I set default values so that anyone can run the pipeline without having to know what they mean, but if someone understands them, they can very easily change that here. I'll just leave them as they are. And the last parameter is the steps of the core pipeline to skip. For example, you may select these 3 steps if you want to skip quality control, but for the purpose of this demo, I'll leave everything unchecked so you can see how it all runs. Alright, so now let's click Generate Pipeline Commands. And here are the 2 commands generated for the CLI to access the pipeline. And I'll show you over here where the conditions are. You'll see that the conditions are now properly formatted. If you remember, we wrote "here they are" in the input, and that was transformed. And that's the same in the analysis command. So let's scroll over to where the model organism is, and you'll see that it'll say Dririo instead of zebrafish, which is what we put. So ChatGPT effectively handles all the inputs. Alright, so if we run the setup command, it'll create the base directories and initialize the structure. And that's just going on right now. Alright, so that's done there. And now I'll run the analysis command. So that's just starting up now. You'll see in a second the output generated. Alright, so metadata is created, and next we have boxplots and the sample correlation heat map, PCA plots, and now it's processing all those pairwise files. So it's telling you the amount of significant genes, how many are upregulated, how many are downregulated, and it will do that for all 20 comparisons and then generate a summary. And so all these outputs are being saved to cloud storage where you can later download them, and I'll show you that at the end of this command. So the comparison files are processed. We have MA plots, and next are the volcano plots. And the next part here is ortholog mapping. So we're making an API call to an external resource. So this part may take a few more seconds. Okay, and the ortholog mapping is finished, and now we've got a heat map as well. And so now we're going to move on to the enrichments. So the Go enrichment and then the KEGG enrichment. So it'll do all the enrichments for the comparisons of interest, and you can see how choosing all 20 of the comparisons would lengthen this process, although it can be done, and that's why we're just doing them with 4 comparisons. Okay, and now we're moving on to the KEGG enrichment. And this is the last step of the analysis now. Okay, so that's finished as well. And now the command's complete. So now let's take a look at what we got in the outputs. So these are all the different outputs, the files that you saw being generated. And at the bottom, you'll find a spot where you can prepare a zip folder to download them all. So I'll do that. Right now, they're all being prepared. And now we have that zip file. So I'll go download that. And you'll see it's being saved here as pipeline output. Okay, and now I'll navigate to that file and unzip it. And right now they're all being extracted. So you'll see the pipeline output folder pop up soon. Okay, here it is. So you can see the subfolders, data, plots, and results. I'll go into data and show you the metadata first. So I'll pull this over. This is auto-generated from all the different conditions and replicates that we gave the pipeline. In the plots, we have the box plots. They're all there. And then you have the correlation heat map, so I'll show you what that looks like. And this is all auto-generated. And then you saw the other plots we have. I'll show you the KEGG enrichment bar plots. So here they are. Just pick one to show you. These are all the pathways that are significantly expressed and this is just one pairwise comparison of interest, but they're all here. And then in results, you have access to the raw CSV data that the plots are created from. So if you want to verify the results or have someone look at the data for manipulation and further analysis, that's all here. So here are all the different orthologs and their expression values. That's all there if you need it. And again, you have the same data for the enrichment plots as well. So they're all here, all the CSV data. So as you saw, the pipeline is able to generate all those outputs in just a few minutes with very minimal input and technical experience. And this low barrier to entry will help Canurta speed up their preclinical trials and bring their therapies to market as fast as possible so they can help more people. All the code for this project is open source and you can find it in my GitHub repository. If you want to use this workflow, you can deploy it using an app like I did with Streamlit, or you can run it all from your terminal. Thank you.